Viruses and bacteria are cyclical in the body. The body is on a cycle, therefore viruses are on a cycle. Just like the weather is on a cycle, so too is the body. When toxic accumulations reach its highest point in the body, the bursting tide can no longer be held back. This may occur due to seasonal changes, climate changes, and temperature changes, stresses, strains, and worries. The changes in the seasons signals the body to dump large amounts of toxin accumulations into the blood. When toxins enter the blood system, bacteria must proliferate in order to break down and eliminate the toxicity now in the blood. However, if the microbes become poisoned to death due to the toxic nature of what is in the blood and what has accumulated in the body, the cells will create and manufacture viruses that are non-living protein solvents. They are protein structures. Viruses come in and break down the matter. They eliminate it so that the body can restore homeostasis. Without viruses, the body would have no recourse to cleanse itself and would die of toxicity. The nature of viruses must be understood. Viruses are not alive. Viruses are not contagious, and I will explain why. Viruses cannot be transmitted between people. Viruses originating outside the host cell cannot infect another host. The virus will not have the necessary RNA or DNA to infect other cells. Cells will not be infected by foreign debris, and that is exactly how a foreign virus is treated by the body. Because the body cannot recognize the virus, since it does not have the key or time to its activity. As a result, the virus is treated as foreign debris. When you inject animal viral tissues, the body does not recognize it, because it did not originate within the cells of your own body. Likewise, if a virus is transmitted through the air somehow, the virus will not be recognized in this way either. We also have the functions of the mucous membrane. When mucus is observed under a microscope, it exhibits a weave pattern. This weave pattern helps filter out foreign debris. If I was somehow able to manufacture viruses in a lab by the trillions, enough to be able to spray it on a large population, this would still not spark the virus that I intended to create in the victim. However, if you are breathing this mist in, it is a foreign contamination, which the body must break down. In those cases, if it is toxic enough, the body will manufacture a respiratory virus. This is in order to break down the toxicity and eliminate it. If we look to China as an example, we can see that the air pollution there is so toxic that people have to wear masks in order to protect themselves. You can imagine, after breathing this in for an extended period of time, you are going to develop a respiratory illness. It just so happens that coronavirus is a respiratory virus. Coronaviruses have been around forever. If you have ever had a cold, it's likely that you have had a coronavirus at one point or another in your life. Coronaviruses are not new. However, the media has hyped it up and tried to convince the mass ignorant sheeple that it is new and dangerous. If we look to Italy as an example, we can see that Italy has one of the highest pollution rates in all of Europe. Therefore, you have a population there that is going to detoxify their bodies, and they're going to do this all at the same times. As I explained, viruses are cyclical because the body is cyclical. Toxic accumulations take time to build up, and they build up in the majority of the population at the same time. Therefore, the majority of the population will release their toxic accumulations around the same times as well. This behavior is what I call the illusion of viral activity. This can be exploited by those in power because the person that is observing this believes incorrectly and out of ignorance that it is contagious. But this is simply not the case. The study of viruses is flawed by nature. In order for them to be observed in laboratories, viruses must be cauterized and taken and dried. This is called irradiation. This also takes the moisture content out of the virus. This is an attenuation process that kills the virus. However, as stated, a virus is not alive. Usually, it is animal viral proteins that are used to conduct these observations. Viruses are observed in petri dish environments that are sterilized. Toxic serums are used to keep the cellular activity alive but not healthy. Cells in such an environment do not have at their disposal the full range of microbial cleansing agents as it would appear in the human body. Therefore, scientists will always observe the prominence of viral activity, because cells must use viruses to cleanse themselves in such a toxic environment. They do not have bacteria or parasites, which they can call upon to consume and eliminate this toxicity. Therefore, scientists observe that viruses dissolve their own cellular activity, but this is an illusion. In order to observe viruses, viruses must be stained, which causes inaccurate observations of the virus, since the virus is not acting in a natural way as it would in the human body. Science knows this, but continues the fraud and deception anyway. Viruses are so small that they are the size of 0.1 microns. This requires incredibly powerful microscopes to see. Optical microscopes must require the staining process of viruses in order to show up on the microscope. So what does the virus behave like in the human body? The only way we will know that is if we view them in the living tissue. And we have just that, because Royal Rife invented his own microscope that could view living tissue in images up to 30,000 times their size. He was able to view microorganisms in their living state within the tissue. Rife observed that viruses and bacteria only occur due to imbalances within the bodily system. In short, he concluded that viruses and bacteria are pleomorphic, thereby confirming the findings of Antoine Béchamp in the 1800s. Rife states in his findings, quote, "In reality, it is not the bacteria themselves that produce the disease, but we believe it is the chemical constituents of these microorganisms acting upon the unbalanced cell metabolism of the human body that in actuality produces the disease. We also believe if the metabolism of the human body is perfectly balanced or poised, it is susceptible to no disease," unquote.
What anybody will discover when they search the record is that Royal Rife invented a microscope in the early 1930s, which was called the universal microscope, and its unique quality was that it was able to view viruses in their living state, able to view microbes as small as viruses in their living state. Now today, and even then, we had the electron microscope, which can see viruses, but in a snapshot approach. They're dead. They have to irradiate them with electrons, which is the method of operation. It kills them, and therefore you're not seeing the life process. And with optical microscopes, stains are used to bring out features, and the stains themselves are toxic, and once again you've got dead samples that you're viewing. Well, Rife saw this limitation in microbiology and microscopy and decided to address it. So his answer was to invent a microscope which used light to stain the sample, and thereby not killing it. And he was able to, using this method, obtain extreme magnification, extreme resolution, and he was able to view his samples in the living state. So he could see viruses moving around, going through their processes, which you can see with an optical microscope, but it was his ability to see viruses, which in the living state, which was his key innovation. And all of his other inventions, and there were many, all related to this, sort of bounced off the principles that the microscope operated on.
Viruses cannot cross species for a number of reasons. For one, RNA and DNA of the animal is not compatible with human RNA or DNA. According to science, they claim that animal viruses can be transmitted to humans. But the human body does not manufacture and replicate animal cells and tissue. We are humans, and when we eat animal flesh, it is converted in the stomach to human tissue, which we use for our own needs, to build muscle, for example. The only way to get animal viral tissue into a human body is through injection. Only then can animal tissue be observed in the blood. Therefore, the supposition that AIDS came from a monkey, or that coronavirus came from a bat, or that Ebola was caused by a bat, this cannot occur in nature. Now ask yourself, why is it that only in the past 40 years have these viruses occurred in nature? Why didn't they occur 1,000 years ago? Why didn't they occur 200 years ago? These viruses would have occurred in nature long before now, if what science is saying is true. It is impossible for animals to transmit viruses to humans, and it is impossible for them to transmit it between themselves. It is also impossible for humans to transmit viruses between themselves. The body does not recognize external viruses. Viruses have to be created by a cell in your own body and contain the necessary RNA or DNA given by your cell. There is no such thing as a majority viral strain. Every body is going to have a different strain because the body creates the strain for its own usage. Quoting from The Poisoned Needle by Eleanor McBean, 1956, nutritional and medical doctor. Quote, "During one of our most widespread polio epidemics in 1949, contraction of polio by definite contact with other victims of the disease was not established in an elaborate study made by the New York State Health Department. The United States Public Health Service in its studies also found the same negative answer during succeeding outbreaks of polio. In short, they learned that the disease was not contagious." Time magazine commented on this unexpected revelation of these surveys by remarking that, quote, "when and where people catch polio remains a mystery," unquote. The supposition that viruses entered through the mouth and nose led the experimenters to make exhaustive tests on both animals and human beings, mostly prisoners and orphans in institutions. They were exposed by close association to polio patients, and their throats and nasal passages were frequently swabbed with matter from the patients that was supposed to contain viruses. No polio was produced in this way, and about the only noticeable after effects was that the subjects of the experiments lost their sense of taste and smell due to the damaging effects of the poisons in the serum that was used on the swabs." Unquote.